Mr. Chancellor, it is my pleasure to present to you Professor Randall Prather, holder of the Distinguished Professor of Reproductive Biotechnology at the University of Missouri. Internationally renowned for his studies into early embryonic development, his transgenic work with swine led to the first cloned pigs in the 1980s and formed the basis for much of the animal model work that is now considered fundamental to human health and fertility. En plus de ses centaines d'articles de recherche, il est régulièrement invité à partager ses idées partout dans le monde. Il a déjà fait plus de 100 000 contributions à GeneBank et il est l'auteur de nombreux brevets. Son financement à date est impressionnant, ce qui représente presque 80 millions de dollars. Cet incroyable dossier d'aide à la recherche et de réalisation est dû en grande partie à la démarche de toute sa carrière de chercheur privilégiant la collaboration entre sciences animales et humaines. Professor Prather serves as Associate Leader of the Food for the 21st Century Reproductive Biology Cluster and is a co-director of the U.S. National Swine Resource and Research Center. In 2011, he received the Distinguished Scientist Award from the Christopher Columbus Foundation in Washington, D.C. Monsieur le Chancelier, je vous présente le professeur Randall Prather pour que vous lui confériez la grade de docteur des sciences honoris causa.
Ladies and gentlemen, it's my great pleasure to invite Dr. Randall Prather to deliver the convocation address. Dr. Prather.
Well, thank you very much. This recognition is truly a surprise and an honor, especially from an institution like McGill, which is considered by many to be in the top 25 in the world, maybe top 5. I'm currently at the University of Missouri in Columbia, Missouri. We too have an agricultural school, of which I'm a part of. We have a medical school and an engineering school. We have a veterinary school on campus, but you guys have access to that. You have an incredible resource here at McGill that putting people together from all these different institutions, these different expertises, can really create some interesting things, spanning disciplines to create knowledge that will positively affect people's lives. You should be proud to have graduated from McGill. It's a great place. Because this is truly a great place, I'm very honored and humbled and delighted to see that I have the recognition from the work that we've done at the University of Missouri. I'd like to make 3 different points. Point number 1 is be open to working with others. Point number 2 is take advantage of the situations that you're given. And point number 3 is find support. I found both a cheerleader and an advisor. So, back to working with others. One of our most notable accomplishments was that we were the first group to knock out a gene in a pig. Since that time, we've now made 40 different genetic modifications in pigs. That first pig cost hundreds of millions of dollars and took many years to create. Now the technology is available to create that same pig within about 5 months. One of our most notable accomplishments was to create pigs that have cystic fibrosis. This is caused by a mutation in a gene called the CFTR gene, or the Cystic Fibrosis Transmembrane Conductance Regulator. It's a chloride ion channel that's expressed in epithelial cells in the lungs. A mutation in both copies of this gene results in a mucus buildup in the lungs and subsequent infections. In North America, about 5% of the human population are carriers of a mutation in this gene. We have, 500 people here, maybe? 25 of you are probably carriers, and you may not even know it. 70% of the people that have cystic fibrosis have a specific mutation called a delta F508, which is a deletion of phenylalanine, which is the 508th amino acid in that protein. The ability to introduce this mutation in mice has been around for about 30 years. However, when this mutation has been introduced, the mice get none of the symptoms associated with cystic fibrosis, other than they do have a defective chloride ion transporter. Incidentally, the symptoms of cystic fibrosis include more than lung disease. Humans are born with meconium ileus. This is a mucus plug in the intestine. If it's not corrected, it will result in a perforated intestine and death. But that's not all. The gallbladder is congealed, the bile ducts blocked, the pancreatic ducts blocked. Those of you in nutrition may know that some cystic fibrosis patients take pancreatic enzymes orally with their meals because they can't get it from their pancreas. Liver lesions will develop, and males are often infertile. In collaboration with Mike Welsh at the University of Iowa, we created pigs with the exact same mutation that occurs in people. And yes, they show symptoms of lung disease and all the other symptoms of cystic fibrosis. Finally, the physicians have something to experiment on that exhibit the disease phenotype. Before these pigs were created, they could only perform limited experiments on children that had cystic fibrosis. My point in telling this story is that we at Missouri we could have made the pigs, but we couldn't have adequately characterized the phenotype without the group from Iowa. In fact, most of the projects that I work on are projects that are somebody else's idea, not mine at all. So I challenge you to be open to working with others that are smarter than you, because together you can easily accomplish goals that alone you couldn't possibly accomplish. The second point that I want to make is that in whatever situation you find yourself, take advantage of it. While I was a postdoc at the University of Wisconsin, I worked with mice and cattle, and my goal was to work on cattle embryo development only. Well, I was also an undergraduate at Kansas State University, and I had a crop science teacher that was discussing planting corn or grain sorghum. Since Kansas is prone to drought some years, the corn crop didn't make any money, and so there was nothing to pay the banker in the fall. But people like to plant corn, and in those years where it did rain, they could go to the coffee shop and brag about their yield, and that they worked with a nice, clean crop. On the other hand, if you planted grain sorghum, since it's drought tolerant, you could take a check to the banker most every year, but the grain sorghum's a dusty, dirty, nasty crop. So in Kansas, men grew corn. Other people worked on or grew dirty, dusty, nasty grain sorghum. I would argue that the same attitude exists in the livestock industry. Men raise cattle. Others work with those dirty, dusty, nasty pigs, even though on average the pig farmer is going to have a check to take to the banker more often than the cattle farmer. So I was not interested in pigs at all. My undergraduate advisor even forced me to take his swine science class as a senior. And I remember telling him quite explicitly, I was never going to work with pigs, so why bother? When I got to Wisconsin, there was a lot of competition for the resources to work on cattle. We had published the first paper on cattle cloning by nuclear transfer, and I was interested in seeing if it would work on other species. That's when the only person that was working on the pigs in the lab got a job and left. Suddenly there was a vacuum, and I decided I didn't want to compete for the cattle resources anymore. I became a pig embryologist, and we cloned the first pig. This is one of the best things that happened in my career, as I later discovered that pigs were great models for many human conditions. Now I could apply for funding from both the United States Department of Agriculture for agriculture purposes and the National Institutes of Health for biomedical purposes. So keep your eyes open and take advantage of the situation you're given. My final point number 3, is to find support for your efforts. I'd like to highlight 2 people. The first is an 87-year-old veterinarian. He'll be 88 actually very shortly. At the young age of 65, he retired from the College of Veterinary Medicine and came to spend 2 weeks in my laboratory. He never left. What a wealth of wisdom he has been to me and to everyone in the lab. He is the harmless old man that everyone goes to with their problems. He can give advice that I, as an employer, could never give. For example, you need to break up with that guy. He's not good for you. A happy home life makes for a positive attitude and a good work life. In contrast, if home life is bad, then often there's little focus at work. He inspires lab members to become their best. He also sees potential problems with personnel interactions and heads them off before they become disruptive to the function of the laboratory. I encourage everyone to find an older mentor and then, over time, become that older mentor. Incidentally, Clifton Murphy still performs our surgeries. We average over 2.5 a week. And again, he'll be 88 soon. Finally, we all need cheerleaders. I do have manuscripts rejected. I do have proposals that are not funded. I joke that I only write grants because I expect them all to get funded, but in reality, I do write many proposals that don't get funded. In academia, you need to have a very, very thick hide. Criticism comes from students, parents, administrators, colleagues, anonymous reviewers, and the public. I have someone that for the last 34 plus years has been my cheerleader. She's been a constant support during the good and the bad. She's sitting right there cheering me on as I speak. My wife and the mother of our 7 children, Jamie. So to close, I want to encourage you on 3 points. Point number 1, be open to working with others, especially if they're smarter than you. Number 2, take advantage of the situation you're given. And number 3, find support. I found a cheerleader and an advisor. Thank you.